Hello and welcome to this Chemistry World webinar. I'm Ben Valsler, Chemistry World's digital editor. My role today is to introduce you to the platform that we're using, tell you how to ask questions, engage and so on, introduce our speaker and then sit back and along with you thoroughly enjoy the presentation. Today we're learning about molecular modeling for the medicinal chemistry toolkit. We have a fantastic speaker with us today who is exactly the right person to talk you through how all of this works. And my doorbell has just rung, which is absolutely typical. So what we're going to do today is we'll discuss the molecular modeling tools that are geared towards medicinal chemists, and we'll demonstrate docking and enumeration of lead compounds in a drug discovery case study. Now, the key thing, of course, or a significant goal in the design and optimization of drugs is the reliable prediction of structure-activity relationships. Whether you prefer to design your ligands in 2D or 3D, it's extremely useful to be able to predict how your ligand modifications are likely to impact protein-ligand compact structures, and molecular modeling and docking tools are ideal for helping to provide those useful predictions. Now, all of that you've probably read on the page already, so let's get into the beef of how you really interact. Now, the key thing that you will need to know in order to ask questions is in that GoToWebinar panel, which is likely to be down one side of your screen, you'll see there is a box at the bottom, there's chat or questions, depending on exactly what version you're running. At any point throughout the webinar, you can ask us any question you like in there, whether it's about the technical content that you're seeing on screen, how you might be able to apply it to your own work, or if you just want some further information. Any question you have, get it in there at any point, and we'll come to you for Q&A after the presentation. So that's the best way to make the most of this online interactive experience, is by asking all of the questions that you have as soon as they occur to you. Now, we will have webcams for some of today's presentation and not for other bits. If you're on a slow connection, you can, in the GoToWebinar software, just switch those webcams off. You'll still see the screens and you'll hear what we're saying. But if you are on a slightly iffy Wi-Fi connection or something like that, then that's the best way to make sure that you still get to see and hear everything that you do need to see and hear. Now, speaking of that, we don't share slides from Chemistry World webinars. We believe that what's important is not just what you're seeing on screen, but also what's being said. So rather than sharing the slides from today's presentation, we will send you a link to the recorded version where you'll get everything in its full context. So that will be in your inbox some point over the next couple of days. And for those of you who have attended live, you'll also get a certificate. That's our little way of saying thank you for attending the webinar live and really making the most of the interaction that's available to you. So I think that's everything from me. I'm now going to hand over to our guest speaker. Now, as I said earlier, she's ideal for this because not only is she a senior scientist and education specialist at Schrödinger, that education specialist aspect of it means that she really knows how to get these messages across. But Abigail is a registered pharmacist. She completed a master's in pharmacy, followed by a PhD in computational and medicinal chemistry at the University of Nottingham. Her research during and since is predominantly focused on G protein coupled receptor modeling, docking, and molecular dynamic simulation in order to better understand the structure activity relationships. So not only has she walked the walk, but she can also talk the talk. So Abigail Emtage is our guest for today's presentation. She should be joining us now. We should be able to see her and hear her very soon. We can see her screen, which is a good start. So let's get her webcam up and see if we can hear her. So Abby, thank you for joining us.
Thanks, Ben. Hi, can you hear me okay?
I can hear you perfectly, yes, thank you. So once again, you're happy to stick around for questions at the end. So do ask questions at any point as you go throughout the presentation. But for now, I will sit back and let you enjoy Abigail's presentation. Thank you for joining us.
Thanks so much, Ben. So hi, everybody, and thanks to the organisers for the invitation to take part in this webinar. As Ben mentioned, I am in fact a pharmacist by training, and studying medicinal chemistry in the context of pharmacy and being able to look at three-dimensional models of ligands and receptor interactions is what initially inspired me to pursue a PhD in computational chemistry. All this to say, I'm really excited about this webinar series, and I know firsthand that molecular modeling and visualization plays a really huge role in inspiring future and current scientists and enhancing medicinal chemistry projects. So let's have a look. So as mentioned, if like me, you've ever been part of a ligand-based drug discovery project or medicinal chemistry team, you may be aware that chemical information is often shared in many forms, from smiles strings to hand-drawn sketches to lab books or via lists of binding affinity data. So today I plan to spend the next 40 minutes or so discussing the ways in which you can easily incorporate modeling tools into your medicinal chemistry projects. So just to kind of cover what I'm going to talk about today, and as the abstract discussed as well, a really common and important goal is being able to rationalize structure-activity relationships. And whether you prefer to design your compounds in 2 dimensions or 3 dimensions, it's really, really useful to be able to predict how your ligand is going to interact with the binding pocket of your protein or DNA, RNA, so on. And so today, yes, we're going to demonstrate how you can do this with various different molecular modeling tools. So I'm going to split this, this presentation into 3 main sections. So we're going to start today by talking about ways that you can streamline your workflows by using our integrated software solution called LiveDesign for docking, ligand design, and optimization. Next, we're going to look at Ligand Designer, which is a tool within Schrödinger's Maestro visualization software that can enable you to intelligently design new ligands and optimize your leads using protein binding pocket ligand information together, and you can sort of generate things down reaction-based pathways that will be really useful if you're then planning to synthesize compounds. And then finally, we're going to look at how you might want to accelerate or skill up your own abilities in terms of development and learning with Schrödinger's online courses that we're currently offering. So let's start by looking at the first of these, so streamlining workflows with LiveDesign. So I think it's really common for many of us that have been involved in drug discovery and drug design to sort of initially think of drug design as this linear sort of process whereby different teams pass information along, and you may well have been, you know, a medicinal chemist or a synthetic chemist in this lineup, and you kind of expect to have information supplied to you, and then it goes through the testing process, and you come out the other end with a drug candidate. However, as anyone who has done this knows, this process, of course, is not linear. It's very much cyclic, and you go through this iterative workflow. Realistically, each part of this cycle then has its own cycle that involves input across different teams via meetings, data exchange, documentation, many different types of software and hardware even to discuss and disseminate this information. And of course, there are many people involved at every step. And again, each of these people may then be involved in their own research projects or various different projects, which may or may not overlap. So there's a lot of data exchange here, a lot of information that needs to be captured. And this can be then really challenging for big drug discovery teams. So again, there's many things that can go wrong within this process. For example, disconnected applications, teams sharing information in a variety of formats, like what we saw right at the start there. Chemical information can come in many different forms. Again, then you might have information from biologists, from modelers, and everything needs to sort of feed in and be shared. But in fact, it can be really easy for a project to fail or miss vital information for non-scientific reasons. And if the information can't be organized appropriately or stored in a really accessible and dynamic way, then projects might suffer as a result and lose time and lose data as a result of this disconnect. So this is where LiveDesign comes in, Schrödinger's enterprise informatics platform, which I'm going to be demonstrating in a moment. And this can really assist with drug discovery and drug design projects. So it provides a simple platform for each team to work on their own data, and they can then share it with others. And it's really designed for getting past these non-scientific hurdles of drug discovery and sort of circumventing them. So again, it really allows the researchers to access various kinds of data in one place. And in fact, you can see from this slide that this data then feeds in all into this one interface. So the idea is here that we have our computational modeling, we have our chemoinformatics, and we have our experimental data all in one place. And design can be done as well as presentation and collaboration on a project. So it's a really robust way to collaborate and share information within a team and across teams. And again, this is something that, you know, obviously all drug discovery projects need and would benefit from. So how does LiveDesign work? What actually is it? And a really good way to think about LiveDesign is that it's similar to a chemically aware spreadsheet. And we refer to these as live reports. So I'm now going to switch off my webcam and go into the demo and show you what LiveDesign looks like. And we'll work through a bit of a case study. Okay, so going to LiveDesign. So this is what LiveDesign looks like, and you can see here there are many different rows with our various compounds, and they have various different bits of information associated with them. So for example, there might be their name, their Chemical ID in this case. We also have the structure here. We have some information about the binding affinity, as well as some additional information as well that we've added, for example, molecular weight, number of hydrogen bond acceptors and donors, and so on. So again, all the kind of information that you might see in a typical drug discovery spreadsheet, but you can see that this information is in fact live. It's very much like a tab of a browser. It's web-based, so it's constantly updated and constantly live. So we can view these compounds as rows with their populated columns of data. And the live report can range from assays, attached files, calculated properties, user-generated formulas, annotations of all different kinds. So there's all sorts of different things that we can put in these various fields. So I'm just going to go and show you sort of what this might look like right from the start. So if you don't have a live report and you want to create something right from the beginning, what we're going to do here is look at a brief case study of the adenosine A2A receptor. So if we have, for example, an ongoing drug discovery project with the adenosine receptor, and we want to design some new compounds around a known core, and then dock those compounds or use modeling tools to assess those compounds, we can do a lot of this all within LiveDesign. So the first thing we might want to do is add some compounds. For example, we might have a known lead or a known core for our target of interest. So here in the sketcher, a sketcher will come up, and I've just drawn a benzimidazole here. We can then use this or add this. Again, you can certainly modify it however you might like, just via this sketcher tool. We can add this design to our live report. So this just comes in that we're getting the very first structure into our live report, and we can see there that it's now got some extra information. So it's got its ID. Again, if we want, we can include a sort of additional rationale here. We can also include the lot scientist that potentially has synthesized this compound. We don't need this particular column for now, so we can hide it. The same with the rationale, we can just hide that column, and then we can potentially sort of think about what we might want to do next. And there are many different things that we can now do in order to expand this this particular compound, and let's see if we can sort of enumerate this further and get some additional sort of substructures and information. So what we can do now is go to our advanced tab, and we can actually either enumerate it via R groups or a reaction-based enumeration. We can also use a filter to filter sort of specific queries. And I'm just going to go to my pre-run report here, and this is just what we've added here is compounds that all fall within a particular molecular weight, so they don't go above 1000 Daltons, and this this is then just, we've enumerated this pathway in that we've just generated a lot of compounds that all have this same or this similar core structure based on the benzimidazole. So we can generate these types of sort of compounds, and again, if we have some additional adenosine A2A activity, which in this case we do, this is something that we might have imported, or we can again find this information from chemically active databases online, then this again can be imported along with, for example, things like Chembl IDs. So again, I'll just quickly show you that. I don't think I showed you that thoroughly, apologies. We can import various different information here. So for example, Excel, CSV, spreadsheets, and so on. Even three-dimensional structures here, such as Maestro files. So this is a quick way to get information into our live report. And then the next thing we can do is sort of generate some hypotheses and add some additional information. For example, we might want to just briefly sort or color these columns. So we can, again, sort just like you would in a normal spreadsheet, sort by ascending or descending. We can have some nice coloring rules in here. So for example, for really good binders, we might want to color this sort of green, and then say, you know, for less good, we'll have it going through to red. And then we can color in and see that we're getting, you know, some sort of a nice overview there in terms of how our data looks. So the next thing we might want to do is kind of run an SAR analysis and say, well, what different side chains have we got going on here? What different R groups have we got off this kind of core benzimidazole type structure? And what are we seeing that's different? What are we seeing that's similar based on our binding affinity data here? So what we can do is go to the analysis tools section here of our of LiveDesign, and we can either draw in a scaffold. So again, we could draw in our benzimidazole again, or we can actually drag a known structure to this area here, and then just modify elements of it and kind of decide which elements are going to be defined as R groups. So in this case, I'm just going to kind of take off the larger portions of this compound, and I'm going to just replace some of these things with R groups so that we can see here. In fact, I'm going to just put R groups in these positions as well. So anything that then has an R group could potentially be, of course, a different substituent there. So again, what we can do with this is then create this scaffold, and we will then see that our live report becomes populated with all of this information. So we can very quickly see we've got this nice core, which is now nicely shown in our diagrams here in pink. So this is based on our initial core part of the scaffold. And then we have our various R groups, which again are color coded around the way that we chose them in the initial scaffold structure. So again, we can see R1 here is coming off the nitrogen, R2 again, and R3, R4, R5, and so on. So we can see there where they are, and then we can very just easily and quickly look through the data and say, right, what are the really common substituents at these various R groups? Which of the compounds match the scaffold, which don't, and so on. And we can see also that things have been aligned as well based on this core. So it's really easy to just quickly compare our structures now. So this looks good, and again, hopefully, you know, this, you can see the advantages of this, that it would be quite nice and easy to share data in this way and easily generate sort of some nice information about the various R groups in a drug design project. But what would you do if you want to, say, now dock these compounds to the A2A receptor? Now, there's crystal structures for the adenosine A2A receptor. So if you have a molecular modeler in your research project or in your group, then they may have generated a molecular model, and they may be then doing some docking. But rather than doing that in a very sort of isolated or separate way, that's something that can actually additionally be incorporated into a live report. So what I'm going to show you now in this demo 3 area is we have got this docking model has been loaded in. And again, we can potentially go over that at the end, how you might incorporate a docking model into a live report. That requires that you've pre-generated it. So again, we can use this docking model, and then what will happen is if we click on this, the docking will automatically be done with all of the compounds that you select within the live report, and you can then visualize that right in the web browser, so right in LiveDesign here via this small PyMol interface, and we can see immediately, you know, what types of interactions this compound is making with the binding pocket. The dotted yellow lines here represent the hydrogen bonds, and we can see that again this is taking up sort of a good amount of the binding pocket. There may be some additional space here for growth, and we may then think, oh, okay, we can design a little bit into this pocket. We might want to be able to build out some interactions here. If you prefer to look at things in 2 dimensions, another component that is generated as part of the docking within LiveDesign is this nice 2-dimensional ligand interaction diagram. And if you're familiar with using Maestro, you may have already come across these and generated these types of nice visualizations so that you can see very easily there which amino acids, which residues are interacting with your compound and sort of whereabouts they are in relation to the ligand. So there's lots of different tools that can be used to do this. And then another thing we might want to do, of course, is to generate or design a new compound. So what we can do is drag one of our good compounds. So again, here we've got a nice compound which has got really good affinity. And we might say, okay, well, we've seen in the binding pocket that this benzyl ring here seems to be quite deep within the pocket. What can we do to generate some additional new compounds in this sort of area? So what we might want to do is add a methyl group here, and we can add this idea then to our live report. So this compound has now been added. It gets added, I believe, to the end of the report. Oh, it may already be in. Let me just find it. Where's it gone? We'll draw another one and see if that's in there already as well. Okay, so that has been added to the bottom here as well. So we have our 2 new compounds. Because we've added them afterwards, they're no longer showing the same kind of core structure that we've seen previously. But what we can see here is that the docking has not yet been run. So for the compounds there where we see view in 3D, this means the docking has already been completed. In these contexts, we haven't yet run this docking. So what we can do is just say, yes, we want to run this with our A2A model that has been previously loaded into LiveDesign. And we can then just automatically, anybody that is involved in this project can run the docking for a new compound that they've designed right there and then. So this docking will run, and then we'll get the same kinds of visualization and results that we've seen previously. And again, this is just using my browser, so it's taking a little while to load, but we can run this docking almost instantaneously and then look at the receptor ligand interactions. So there's a lot of capacity to generate new ideas within LiveDesign, both in 2 dimensions by looking at the docking in 3 dimensions, and then actually running the docking within the live report. So I'm going to skip back to the slides now. Again, just to say that actually you can really view your data in many, many different ways within LiveDesign. So you can view in panels, in tiles, in this spreadsheet kind of format. We can also generate things like customized charts and forms. And there's obviously a lot more that can be done. And we have a lot of training resources for this, because clearly there's a lot that you can do within this platform. You can also set up notifications, which can again be really useful for drug discovery teams when there's a new compound added, or for example, when a chemist has synthesized something successfully, or when there is some binding affinity data added to the project. So this again can be really good in terms of staying on top of new data. And the main thing really here is that LiveDesign is really useful for collaboration between teams, and it really helps computational projects proceed more effectively and more efficiently, giving teams control over their work, avoiding data versioning issues, streamlining communication, and reducing lost time. So obviously all of this is generated so that a drug discovery project can succeed. And we've looked now sort of mostly within the live report space, but I'm now going to spend a little bit of time just discussing what can be done within Maestro, which again, you may be familiar with if you've used any of Schrödinger's visualization tools, quite similar to PyMol. Again, is a really great platform for visualizing three-dimensional proteins and ligands interacting with them. So what we'll do now is have a look at how you might use the information you generated in LiveDesign, such as some new docking poses, and how you might then take that forward and design using 3D methods with Ligand Designer. So the really nice thing about the Schrödinger tools is that they can really be kind of shared amongst these various platforms. So while LiveDesign is literally live and web-based, Maestro is currently a downloadable software application which enables you to get really nice visualization and generate things like high-resolution images as well. So what we can do is actually import or download information from LiveDesign and bring that straight into Maestro. And then we can use the Ligand Designer tool to enumerate or to further design into space between the ligand and the pocket. And we already saw a little bit of space just visually when we were looking at the ligand in the pocket within the LiveDesign visualization. But in this Ligand Designer context, we'll be able to see even more. So I'm now just going to take you to what we can see, first of all, in LiveDesign, and then we will look at this in Maestro. So for example, we can see here now that our docking for this compound with the added methyl group has run. So we can view this in 3 dimensions. And we can see that, yes, there still seems to be space. The compound has successfully docked. So we've generated this additional this compound with the additional methyl, and yes, this seems to be taking on the same binding pose as our previous compounds, which this was built off of. So what we can do now is actually download these types of structures. We can download one of our leads and explore the current information in Maestro. So what we can do is export this as a Maestro format or a MOL2 format file. So I'll export it as an MAEGZ file, and that will download both the receptor and the ligand. So again, you might want to just save this, or you can choose to open it directly within Maestro if you have that set up on your computer. So that's downloaded, we can see that's downloaded, and that will then be something we can open in Maestro. So I've done this earlier and I've opened this for us in Maestro, and we get this representation. And so initially we see this and you think, oh, okay, I'm not quite sure what I'm looking at here. So what you would want to do is style this to be able to visualize this in a more kind of understandable way. And so what we can do is use this quick select toolbar here to select the protein. And we might want to apply some secondary structure here. So for example, we can add some ribbons. And we might want to also look at the ligand, which has been downloaded as part of the Ligand Designer tool. We can include both entries, so both the ligand and the receptor. and we can then have a look at our interactions in a little bit with a bit more space. So obviously LiveDesign, it was nice to be able to see some of these interactions in the PyMol, web PyMol representation, but looking at it in Maestro just allows us a little bit more detail here. Now, the downside with this is that you may find there's actually too much detail, particularly as well if you're not familiar with using these types of visualization tools. Perhaps you prefer to see things in 2 dimensions as well. And there's a few options for this. We can in fact turn on the 2D overlay, which again just reminds us of what the 2-dimensional structure of this ligand is. And additionally, we can use LiveDesign. LiveDesign's tools, which have then included, in this case, we've got some additional water molecules here, and we can use Ligand Designer, which is the tool we're going to look at now. So we go up to the tasks menu, which is how you would start a lot of jobs within Maestro. Go to the Ligand Designer, and we can analyze the workspace. And I'll just make sure the ligand is included there. So we've got both of our receptor and our ligand included. And we would just analyze that workspace. Now, instantly, you can see that there's a difference in the visualization. And this is where Ligand Designer really comes into its own. So the blue cloudy areas here represent the growth space where there is room between the receptor and the ligand. And the pale blue areas represent space where it is buried in the pocket. So the dark blue is solvent exposed and the light blue cloudy area here is buried. And again, that makes sense based on what we saw previously, but it's just a nice sort of representation here. Now I'm just going to hide this big solvent molecule because we don't need to worry about this right now. So we can focus on the ligand and think about where we might want to grow this. So we can see these areas of growth space, and it looks like, again, we can expand potentially a little bit more into this this buried area here. And to display a little bit more information, we might want to include some of the protein-ligand interactions. Again, if you want more still, we can also include things like the protein surface. So again, just looking at that, we can see actually where the surface is and how much sort of room we have in terms of the 3-dimensional space there. So I'm just going to put the ligand protein interactions on, and then we can quickly see a sort of pared down representation of the various side chains that are near to and interacting with this particular compound. And the really great thing about Ligand Designer is that we can sort of identify some nice regions. For example, we've got a potential acceptor here, we have a potential hydrogen bond donor here, and we can just click on one of these and say, okay, let's try and explore the ligand side chain that might be able to interact or make some nice interactions with this donor. And we know this is the buried area of the pocket, and obviously this benzyl ring is kind of ripe for substitutions. So let's say okay to that, and we can explore the ligand side chain. And now what happens with Ligand Designer is that we get these 3 arrows. In this case, there's 3 areas where we can potentially enumerate or expand this compound. So we're just going to pick the nearest one for now and say, yes, let's try and modify this lead compound in order to design into this pocket. Now, there are various ways you can do this. You can potentially set some rules for specific criteria. For example, you might want to limit the number or specify the number of chiral centers or via a heavy atom count and so on. So you can actually have some property filters on there if desired. You can also, excuse me. You can also then enumerate with, again, in these various different areas. So I'm just going to click enumerate here. And what we see down the bottom right of the screen here is that new ideas, new compounds are being generated that will fit into this binding pocket. So the compounds are being generated and they're automatically being docked using Glide, which is Schrödinger's docking software. So the docking is occurring in the background. These new ideas are being generated, and everything that it manages to dock that fits within this particular space then gets added to this list here. So we can see here we've already got a kind of 5 or 6 compounds have been added, and we can start looking through those. So for example, we can see this first suggestion has been added. We've got quite a lot, we've got a larger substituent there. We've also then got some additional substituents that we can scroll through one at a time, and they're still being enumerated. So this is still occurring. We can see that there's several new ideas still being generated and docked behind the scenes. So let's look at, for example, one of these. Okay, so let's have a look at this one to start with. And if we want as well, we can just style this up by selecting the ligand and making sure it's sort of the same thickness there, just for the visualization purposes. And we can actually have a look over here at the right at this multi-parameter optimization grid that we have going on. And there's various different ways that we can then look at or prioritize our substitution. So for example, we might want to try and increase the hydrogen bond donor profile. And we can see here that it's kind of moderate, and this is the sort of the shape we're getting. We can hover over this and we can see the various parameters that we're getting here. So we can see here that enumeration produced 6 poses, 127 failed to dock, and this is likely because this is actually quite a small space. So we couldn't generate any really large R groups off of this benzyl ring. But we've got 6 new compounds out of this, and we can see in turn what they look like in terms of the multi-parameter optimization area over here. And we can see the types of things that we're generating. Another thing that you might want to do here is actually specify which type of multi-parameter optimization you're using. So the default here is the Lipinski, which has these 5 different parameters, but we can generate this to be drug-like, which also adds a chiral center component, or we can limit this to just a 3-term multi-parameter optimization. So just with PSA, hydrogen bond donors, and A log P. Or, you can actually introduce your own. You can have a custom multi-parameter optimization, which again can be really appealing for specific projects where perhaps there needs to be a particular core or particular binding mode, and you can have that reflected in your multi-parameter optimization. So hopefully that's just given you a little idea about how you can design in 3 dimensions. Another thing that you can do with Ligand Designer is actually edit your compounds in 2D. So if you do prefer to design using 2-dimensional Sketcher, then you can do that right here. And again, this is just like we did with LiveDesign. We can potentially, edit, and we can add to or remove various components here, and then just click on Predict Pose to dock that right there in the workspace and in the workstation. So there we can see our user-defined ligand 1, and we can see with our methyl group that has successfully docked into that binding pocket. Again, we can look at the various different workflows. We can look at the various different multi-parameter optimization options and see how this particular compound compares to the various other compounds in our list. Okay, so I've given you a sort of real quick overview snapshot now of LiveDesign and Ligand Designer, and just in terms of these tools, how they can be used for design and for collaboration within a computational chemistry team. Well, a medicinal chemistry and computational chemistry team. That's the point. You don't need to be a computational chemist to use these tools. So I just put my camera back on now and just talk through this last section of the talk. So this is really just to say how we can support you in terms of learning these tools. So there are many different ways that you can learn about the software. For example, within Maestro itself, there's a help menu which will lead you directly to the documentation and tutorials. Many of the panels within Maestro as well have this question mark where you can click on that, and again, it will take you directly to the relevant documentation page. We also have a really nice list of publications to help you generate new ideas, which can be filtered and explored by category. And then finally, I really want to highlight our training portal. So we have a huge number of freely available videos, tutorials, and webinars, which you can subscribe to or stream at your leisure and download in terms of the tutorials as well. So for a lot of these resources, you will just need to create a free website account, which we'll show you how to do later on. But again, there's some really, really excellent resources here. For example, if you want to get started with using three-dimensional tools or visualization in software such as Maestro, then there's a nice learning series here for Maestro specifically, which will just tell you about, how to style compounds, how to download things, and so on. And a really, really great way to really skill up quickly and cover a really broad range of topics is via our online courses. So I'm part of the education team at Schrödinger. We are developing and have developed several online courses, which, as you can imagine, during the pandemic have been really well subscribed to, just in the sense that people are unable to go to in-person workshops and have not been, able to attend kind of conferences where they would perhaps get the kind of hands-on experience that you would want when it comes to learning new skills and new software. So I'd really encourage you to have a look at our online courses. We currently offer a molecular modeling for Introduction to Molecular Modeling in Drug Discovery course, as well as molecular modeling concepts for polymers, for those that may have more of a material science leaning rather than drug design. And again, with these courses, they're all asynchronous, so you can go through them in your own time. They run over 5 weeks per course, so we have specific sessions, because within these sessions, they're actually a dedicated support team from Schrödinger, so myself included and others in the education team, that are on hand to answer your questions and grade the assignments that are associated with the course. And by the end of the course, you get a certificate to say that you've achieved all of the learning objectives set out in the course. So for example, our drug discovery course, we give you free access as part of the course fee to Maestro and LiveDesign. So you actually get access via our virtual cluster, and so you have access to the software, lots of tutorials to work through, a case study to do, and again, we've had lots of really great feedback in terms of how useful these courses have been at skilling people up and getting people familiar with drug discovery tools. So more details can be found on our website, Schrödinger-online learning, at the main Schrödinger website there, and I believe these links will be available in the chat afterwards as well. And we've got some, again, really clear learning objectives are set out on the course website. We also have a real active learning approach in these courses. So there's videos, there's checkpoint questions as you go through the videos. Each of the videos is quite short and succinct, so you're not expected to watch hours and hours of like long webinars, but there's lots sort of to keep you engaged, and they're very interactive. As well as I said, there's dedicated support team who are available to answer any questions and grade your assignments. So as a kind of thank you for attending today, we're really pleased to be able to offer a $50 discount to anyone who's interested in signing up for one of our introductory courses using the following coupon codes. And if you go to the registration page, once you've generated a free website account, you'll be able to see the registration dates and enter the relevant coupon codes there. So again, just a reminder that if you want to register for one of these courses, to see the available session dates, you have many sessions upcoming throughout the summer. You can click on the register buttons on the website, and that will take you to some more information when the next sessions are coming up and so on. Again, really happy to answer any questions about the courses. I'm just going to finish off by saying that particularly for medicinal chemists, people without any modeling experience, we've had a lot of people go through this drug discovery course now, and we've had some really, really excellent feedback. Generally, people have found it to be really, really helpful. And yeah, in terms of skilling up, in terms of Maestro and LiveDesign. So I hope that is interesting to you, and I'd certainly encourage people that perhaps have not been able to attend in-person workshops, webinars, conferences, or perhaps who have new employees onboarding or new students beginning, as a way to just really engage in a sort of very scientifically focused and logical approach to drug discovery, rational drug design, and modeling software. So I just want to finish by saying thank you and sum up by sort of recapping what we've covered today. So automated docking, and efficient team working within LiveDesign, designing into a binding pocket growth space, so either by 3D or 2D optimization with Ligand Designer, and accelerating your development with Schrödinger online courses, learning new skills to equip the whole team. So I just want to say thank you for attending, and I'll be around for questions. Thanks.
Thank you very much, Abby. Fascinating. I'm amazed how much we fit into the last sort of 40 minutes or so. So Schrödinger are a regular partner for Chemistry World webinars. One of the reasons for that, of course, is because they give us excellent people like Abby who are covering quite complicated stuff in an extremely easy to understand way. So thank you, Abby, for joining us, and keep an eye on chemistryworld.com/webinars for other upcoming projects that we have with Schrödinger. There's roughly one a month for the rest of the year where we'll cover a broad range of topics with them. Keep an eye. There's an excellent looking one coming up on the 24th of June. That one's on materials innovation. So if you're more of a materials bent than a drug discovery bent, and that's the one for you. Before we go into the questions though, let me just show you and talk you through that process of getting an account with Schrödinger. So I'm just going to share my screen for you if this will let me. There we go. So we should in just a second see my screen. And this is that link that we shared in the chat and was shared on screen, which is the Schrödinger online courses page. Now, if you have never been here before and you want to sign up for a course, then you will have to register for an account. It's very easy, very quick. I did it in about 10 minutes before this webinar started. But basically here you'll find all of the information that Abby's just mentioned and more. And in particular, those two courses she was talking about that they're currently running are here. There's also some info on upcoming courses. If these aren't quite your area, then there's bound to be something coming up soon. In order to find out a bit more information and register and see those dates and take advantage of that $50 code, if you hit register, it will ask you to sign in. You can see that there is the register for an account button there. So you just click on that, put in the details that you need. You'll get an email confirming your account very quickly, after which you will then be able to actually get into, yep, that is the right page. So if we then go to the molecular modeling course sign up, you'll see here are all of the dates with the registration dates, payment deadlines, everything you'll need to know. Now, once again, there'll be links to this, not only in the chat function now, but we'll put them in the follow-up email. That's the same email that has the entire recorded presentation for today. So if you did come in a bit late, don't worry, you haven't missed anything that you can't see again. That recording will be in your inbox in a day or so's time, along with your certificate. But there you can see we have all of the upcoming dates and the frequently asked questions, a bit more about the course itself, and the registration down at the bottom. And coupon code here, that is where you'll want to put in the code. Now, those codes are valid for a little while, so you do have a little bit of time before they will expire. I've been told they expire on the 26th of August. So you've got a couple of months still to maybe do a bit more reading around and decide that this is definitely the right course for you. Then use those coupon codes to make sure you get them in the best value that you possibly can. So I will just hand back control to Chris. In fact, I'll let Chris take control, so we'll see the links on screen again. Have a look in the GoToWebinar chat, copy out any of those links that you need for now, because that chat will go away at the end of the webinar. So now is the time just to do a quick copy and paste and make sure that you've got hold of them. And the last sort of 10 minutes, let's get through some of these many fantastic questions that people have asked. So there's one, Sumya asked about drawing structures. You've obviously shown us how to draw them in Sketcher. One sort of general question is, can you import those structures if you already have them elsewhere?
Yes, you can. So both in LiveDesign and in Maestro, you can import via either Smiles or any other sort of 1D, 2D form of the structure. Again, there's various different file types that can be accepted and imported into both Maestro and LiveDesign. And if you can potentially draw it in one and then import it into the other. So if you prefer to draw using the Sketcher, you can do that in either Maestro or LiveDesign and then not have to redo it again. You can just import between the two.
So people who decide, actually, we're going to change from what we currently use, this is the right software for us, you don't have to start from ground zero. You can import everything you need.
Yes,
and we had
a
similar question as well about docking models and so on. So can you design, import your own docking models, or do you have to use what's already there?
Yes, you can, and that's one of the really powerful things about LiveDesign. So using pre-generated docking models that, for example, the computational chemist within your drug discovery team has pre-validated or has potentially been using for the project up until this point, you can import that in. So if you're familiar with glide docking, that's the grid file that you would import, and then any additional core constraints information. For example, if there's ligands that are reference points for core constraints, or there's a particular structural file that you want to use. So you can generate those models within Maestro and then import them directly into LiveDesign.
Excellent. Akash asked as well on similar lines, in LiveDesign, can you also get a docking score that will tell you how well how effectively your ligand was binding?
Yes, yeah. So I didn't point that out, but the docking score is actually listed in the live report. So that's there's the panel with the docking visualization has both the docking score, the visualization, the ligand interaction diagram. So there's sort of all the things that you might want to see just generated there for you in the live report. And of course, you can add additional columns if you want into other components. For example, if you're used to looking at specific components of that score, again, anything that is generated as part of the default docking score is available for you to then add as a different column, as a separate field in your live report.
And of course, you showed that you can essentially show and hide those columns. So
yes,
if your particular aspect of the research project doesn't need to know that, you can hide it, but the data is still there.
Yeah, that's a hugely versatile feature, especially because not everyone needs to see the same information, but actually, as you said, it's all there and it will all be there behind the scenes.
Excellent. So speaking of how people actually use it within a team, Hira asked if we are using LiveDesign within our group, can we restrict it to only the people that we want, or is the data that we put in available for everyone?
Yeah, that's again a really good question. Of course, we prize security, and so anybody that you choose to share it with specifically would have access to it, but you can of course keep that private. And each institution or organization that has their own LiveDesign server will be very much private to them, so nobody else can access that data. So certainly amongst the team, that's very useful, because you can, once one person designs the compound and names it, it's then available for anybody within the accessible limit. But beyond that, then no, that server is locked down to you and your team, so nobody else will be able to do that. And again, if you want to generate your own private live reports, then you can do that too and not share them with anyone else, and that would just be your data.
Excellent. We still have about 5 minutes. It's still lots of good questions coming in as well. So if we run out of time, then I'm very sorry in this instance, but we do keep a record of all of these questions. So the Schrödinger team, if there are specific things they can get back to you on, will get back to you. They'll have your details and they'll be able to get in touch. So just because we're running out of time, do still keep those questions coming in. Let's get through some of these others then. Okay, well, talking of more specific things to get into, Cezanne has asked, is it possible to add additional info while designing the molecules like blood-brain barrier permeability, GI absorption, etc.?
Yeah, that's again a really great question. So a couple of features within LiveDesign, you have the option to add your own free-form columns. So this can be anything from, a formula that calculates the binding affinity difference between, say, two receptor subtypes, or it could be as simple as, a field that enables you to add in this additional specific section of information, such as blood-brain barrier permeability. So any kind of field that you want to add, just like a spreadsheet, you can add that as an additional column, as well as link it to other columns if it sort of is relevant to do so.
Okay, Sam asked if you have to optimize the ligand structure before docking.
In LiveDesign, no, this is all done for you behind the scenes. So, and the same with Maestro, in that if you're preparing a ligand and a receptor for docking, you would need to go through the protein preparation process. You would need to go through the ligand preparation process in order to generate the correct torsions and protonation states and so on. That's all done for you in LiveDesign behind the scenes. So it just takes out all of those things are automated. Basically, it goes through the exact same workflow that it would if you were doing it hands-on in Maestro using Glide and LigPrep and the tools that you may be familiar with if you do that kind of modeling, but that's all done for you. So it's made really accessible in LiveDesign. Anyone can do it.
Excellent. Thank you, Sam, for that question. Sticking with docking and so on, Sana has asked, during the course, the training courses, the sort of temporary access that you're given to LiveDesign and Maestro, can people bring their own compounds, do their own work, or is it all on sort of pre-regulated compounds that are just used as examples?
Again, that's a really valid question. So for the courses, because of the way that we provision the resources, we do ask that you only use the course material for the instances that are available during the course. So again, just to stop people sort of running up big disk errors in terms of running out of space, we want to make sure that you're able to actually complete the course content. So we've pre-selected the compounds and ligand libraries that you'll be using, but you do have the opportunity to design new compounds in LiveDesign. So there's it's not just pre-run data, you very much get the hands-on approach, but we give you the protein ligand targets as the starting point. So yeah, so in terms of bringing your own data, no for the current courses, but certainly if you have a Schrödinger account, I would recommend speaking to your account manager about sort of more tailored workshops for a particular drug discovery project or team. That's certainly what a lot of our application scientists will do, will tailor that to you.
Speaking of which, we have had a question about the courses, about the other courses. So could you tell us about the other online courses that cover different topics?
Yeah, so we have a couple that are coming soon. In fact, I've been working for this past 6 months or so on a more advanced course. So currently available, we have our 2 introductory courses, Introduction to Molecular Modeling, Introduction to Polymers Design, and they're really good if you've never used Maestro before, if you're completely new to molecular docking, or perhaps you have, but you want a bit of a refresher. Again, really good sort of level of skills. For some of the courses that are coming soon, for example, we have a high throughput virtual screening course, which is going to be released anytime soon. I believe that the first session will be at the end of June. And that covers a sort of a far more advanced workflow. So it sort of requires that you have some Maestro knowledge already, and we'll be using tools such as GPU shape screening, high throughput screening using machine learning models, through to free energy perturbation prediction with absolute binding FEP. So some really sort of more computational chemistry sort of focus in terms of the tools. So if you're a comp chemist or a medicinal chemist with a real curiosity for high throughput screening, then that again is going to be a really exciting course, I think, for people wanting to really advance their skill sets. And then beyond that, another course that we have in the pipeline is to do specifically with antibody modeling. So if you do a lot of homology modeling and specifically for antibody research, then that course is going to be geared specifically to that.
Perfect. Thank you very much. We have run out of time. I should point out, when you go through that process to register for your free Schrödinger account, one of the questions is a series of tick boxes for areas that you're interested in. So you should be able to pick the specific bits you're interested in, and then you'll get the information, a bit like Abby was just saying, about those particular areas, and you'll find out, I'm sure, about all of those upcoming courses as soon as they're released. So go now and register for your free Schrödinger account, get all the information you need, take advantage of your $50 discount that we're delighted that Schrödinger were happy to bring to our webinar today. So it really is an extra bonus for people coming to Chemistry World webinars. So everybody wins, I'd say. But that is it. We've run out of time. Thank you very much to Abigail Emtage, who is from Schrödinger. Thank you to Schrödinger in general, and to Dan, who's been in the background helping us sort out all of your questions. He attends a lot of our webinars now, and it's really useful to have an expert from Schrödinger in the background picking out what he thinks are the best questions. So thank you, Dan. Thank you to Chris Pink and Francis Edison, who are from my team at Chemistry World, who have also been helping things to run smoothly. So that is it for now. Before we go, just make sure you copy those links and those codes from the chat function, because that will go away. But if you do miss anything and you'd like to come back and watch it, keep an eye on your inbox for the recording. We will also send you those coupon codes and the links in the follow-up email that will come to you soon. This has been a Chemistry World webinar. Keep an eye on chemistryworld.com/webinars to find all of the upcoming webinars we have with Schrödinger and with our other partners, and other things that we like to cover of general interest to the chemistry community. And thank you a huge amount to the 400 or so people who've been in the room with us today. It's fantastic to see such a large audience engaging with such an interesting topic. A final thank you to Abby for being incredibly engaging for the last hour, to you for your questions, to Schrödinger, to my team at Chemistry World. We'll see you again for the next Chemistry World webinar. Thank you very much.